Hello everyone, welcome you all in this video lecture. So in this video tutorial, we are going to discuss about single nucleotide polymorphism. So let's start with the introductory part. So basically, the single nucleotide polymorphism is a condition or it is a DNA sequence variation which occur very commonly in the population. Now in the genetical study or in the terms of biology, a single nucleotide polymorphism is defined as a germline substitution of a single nucleotide at a specific position in the genome. So they are also called as SNPs, SNPs. So before moving to the topic, let's understand that what are the nucleotides and what is polymorphism. So we know that nucleotides are the monomeric units of nucleic acids and they are known as nucleotides. So we know that nucleic acids are of two types, DNA and RNA, and they are discovered by Friedrich Misser. And these DNA and RNA molecules act as hereditary material. In most of the organisms, DNA act as a hereditary material, but in the case of RNA viruses, in these organisms, RNA act as a hereditary material. So now these nucleotides are made up of three components, which are a nitrogenous base, and they are of two types, which are purines and pyrimidines. So there are four types of nitrogenous bases, which named as adenine, thymine, guanine and cytosine. In the case of RNA, the thymine is replaced by uracil. The another component is a 5-carbon sugar, which is named as pentose sugar, and the carbon sugar varies in both the nucleic acids. In the case of DNA, it consists deoxyribose sugar, but in the case of RNA, it consists of ribose sugar. The third component is an ion of phosphoric acid. So these are the components of nucleotide. Now let's understand that what is the polymorphism. So in common language, the polymorphism is a condition or is an ability of a particular organism to appear in different shapes. But in the terms of biology or in the genetic terms, polymorphism is defined as a discontinuous genetic variation which results in occurrence of several different types of individuals in the particular species. So in the genetics, the polymorphism is a condition where there are different individuals of a single species occur due to several variations in the nucleotide sequences. So this condition is called as polymorphism, and a single nucleotide polymorphism is known when a DNA sequence variation occur due to difference in a single nucleotide in the genome. So basically the word single nucleotide polymorphism is made up of three words, where the word single means one, nucleotide means the nucleotides consist of nitrogenous base pairs, phosphoric acid and sugar, and the polymorphism means alteration. So when in any organism of a particular species where in between these two individuals only variation or alteration of particular single nucleotide occurs, it is called as a single nucleotide polymorphism. Now let's discuss that what are the causes of polymorphism. So basically the polymorphism is considered as a type of genetic mutation or it caused by due to several different types of point mutations in the organism. So these point mutations give rise to the different alleles which contain alternative bases at a given position of nucleotide. We know that what are the alleles. So if any organism have two alleles for a particular trait which can be homozygous or can be heterozygous. So these two alleles contain alternative bases for a single nucleotide, and this is called as single nucleotide polymorphism, and it is caused due to several point mutations like insertion, deletion and many other mutations. So let's understand this concept with the help of this diagrammatic presentation. So here we can see two different alleles for organisms of single species. So here in the first diagram, we can see the structure of double helical DNA consisting nucleotides, consisting nitrogenous base pairs. So these nitrogenous base pairs are cytosine, guanine, thymine and adenine. We know that cytosine always pair up with guanine and thymine pair up with adenine. So in this structure we can see here in this position the cytosine binds with the guanine, and in the another picture, which is the picture of mutant allele of the particular organism. So here we can see this, in this place the cytosine and guanine are replaced by thymine and adenine, and all the other nucleotides are similar in both the alleles or in both the DNA sequences. So here we can see in both the pictures only a single nucleotide variation occur, and this is called as single nucleotide polymorphism, because only a single nucleotide altered due to the replacement of nucleotides. Now let's understand about the types of single nucleotide polymorphism. So basically the SNPs can occur in any position of the gene, including coding sequences as well as in non-coding sequences. So previously it was known that in the non-coding sequences SNPs cannot affect any function. But later it was diagnosed that SNPs in non-coding regions can have higher risk of cancer, because we know that the non-coding sequences act as regulatory elements, or they perform all the regulatory expressions or gene expressions. So that's why they cause several types of cancers. The another type of SNP is coding sequences SNP, where the nucleotide polymorphism occur in the coding sequences, and they are of two types, which are synonymous SNPs and non-synonymous SNPs. So in the synonymous SNPs, they do not result in a change of amino acid in the protein, but they can affect another functions in many another ways. And in the non-synonymous SNPs, they have the ability to change the amino acid of proteins, because these sequences are responsible for the translation or responsible for the protein synthesis in the cell. So the third type of SNP or SNPs is called as intervening SNPs, which are found in the intervening regions of particular gene. So now let's understand about the nomenclature of SNPs, or how they can be named. So there are various different nomenclature methods which exist for the naming of SNPs or single nucleotide polymorphism. So here this is the example, this is the pattern for a nomenclature of SNPs. Here we can see where a SNP is named as C.122AT. So here this indicates that a SNP which have C, which indicates the codon, and this 122 is the number of codon, and A and T as the nitrogenous base pairs. So in this naming, this indicates that in a particular gene, the codon 122 is polymorphized or altered, and in this position the adenine is replaced by thymine. And there are many other methods also for the nomenclature of these SNPs or single nucleotide polymorphism. So now let's understand about the applications of single nucleotide polymorphism. So basically they used as the genetic markers for different genomics and genetic research. We know that they found in all the organisms of single species. So they are the best genetic markers used by several genetic or genomic researchers. The another application is these single nucleotide polymorphism used in finding the disease probability. So it is used in the linkage disequilibrium, and it can find out the disease probability in the particular organism, and they can be also used in the haplotype mapping, and they are the type or kind of a genetic marker, and that's why they used in the determination of disease or a particular trait which have the ability to cause a disease in the particular organism, and they have the association with several genetic variations, because we know there are several genetic diseases which caused due to genetic variations or several type of point mutations in the organism. So with the help of these SNPs, it can be found out due to the genetic marking of these SNPs. The another use of these SNPs is for personalized genetic analysis. So with the help of these single nucleotide polymorphisms, a genetic analysis of a person can be analyzed. Now let's understand that how SNPs or single nucleotide polymorphism can analyze in the particular organism. So there are different methods for the analysis of single nucleotide polymorphism in the particular organisms, and they can be used for SNP analysis. So these methods include DNA sequencing, DNA microarray, PCR RFLP, which is a type of polymerase chain reaction, and electrochemical analysis, capillary electrophoresis, and single base extension. So these are the methods of genotyping or different types of genotyping methods which are used for the analysis of single nucleotide polymorphism in particular organism. Now the variation or alteration is in a single nucleotide, or the concept of single nucleotide polymorphism can cause several types of diseases in the organism. So there are different genetic diseases or inherited diseases which can be caused due to the point mutations in the particular genes. So there are diseases named as sickle cell anemia, which is a type of anemia or a shrinked red blood corpuscles, and it is a type of genetic disease. Another disease called as thalassemia, diabetes, heart diseases, cancer, and Alzheimer, and there are several other diseases caused due to the SNPs and single nucleotide polymorphism in the organism. So when the SNPs do not usually function individually, rather than they work in coordination with other SNPs to manifest a disease, such as in osteoporosis, and in the also they can be included.